Okay, lecture 28. We are on the way, so we're going to try to finish up the blood and move on to the nervous system. So this week we're doing the 7th and 9th lectures on YouTube. You can meet in the classroom if somebody wants to project it. You're more than welcome to do that. Review sessions on Thursday. 5 to 7, somewhere in that neighborhood. And then exam Friday the 11th. We'll try to get started at 11, and you can have as long as you want. So again, there's lectures 13, 14, 16, 17, 18, 19, 20, 21, and 22. Format will be multiple choice, matching, and putting things in order. And again, as much time as you need. So this week we're going to try to finish up the blood, how the kidney and the lungs manage pH, and then move on to the nervous system, and then we'll break that up with exam 2 on Friday. Rest of the semester, endocrine and liver and metabolism, running and pulling, work in the body. How does all the things we've talked about come together? Exam 3, last week of classes, immunity, teeth, and the eyes is what we're scheduled to do, more or less. And then we have exam 4 during finals week. So last time we mentioned the Henderson-Hasselbalch equation, where we take a gas, that is CO2 right here, and we have water, which is the solvent in the body. We always think of that as being an excess reactant in this equation. We have an unstable intermediate carbonic acid, which will either go back to carbon dioxide and water or decompose into a hydrogen ion and a bicarbonate ion. This is a key reaction where the lungs are involved, the kidneys are involved, and the cecum will talk about the absorption of VFAs from the hindgut fermentation and how that fits in there as well. So we have this slide. I'm going to come back to that at the end of this lecture. We're going to talk about respiratory versus metabolic, i.e. lungs versus kidney, and where we fit in on some of these other things. So I want to explain some things first, and then we'll come back to this slide. I know it's out of order in your handout, but we'll take it from there. Okay, so we talked about mass action. If I add CO2 at this end of the equation, everything's going to shift over this way. So if I put a whole bunch of CO2 in here, I'm going to generate more hydrogen ions and more bicarbonate ions as the equation, as the reactants and products in solution come in balance. If I add a lot of bicarb at this end, I'm going to push the equation this way. Mass action is going to generate more carbon dioxide and water. So whichever way we go on this equation, we're going to control the amount of hydrogen ions present, and that's how we control blood pH. So this equation, the Henderson-Hasselbalch equation, is key in that controlling of the pH. We've also got strong ions, but that's beyond this class. So if I want to reduce hydrogen ions in solution, I have to get the equation to move this way. So the easiest thing to do, best thing to do, is build a pile of this, the bicarbonate ion, and hopefully that will combine with this and drive the equation this way, removing hydrogen ions from solution, which should increase my pH of my solution. So I can think of it as building piles. So not a tilting seesaw and everything slides, but we need to think about building piles. So if I'm going to build piles, if I build piles over here, make this bigger and bigger and bigger, everything's going to slide downhill. If I make this bigger and bigger and bigger, this lifts up, and then we slide downhill in that direction. So if I add more CO2 here, that means I'm going to generate more hydrogen ions. If I add more bicarbonate here, it means I'm going to take away those hydrogen ions and generate more carbon dioxide gas. So if we do this kind of in steps, we've got hydrogen ions. Here we want to reduce the hydrogen ions. I can do that two ways. I can either decrease this, and that'll make this bigger than this, so the equation wants to run the way the arrows are, or I can increase bicarbonate. I can put more bicarbonate up here, and again, I'm going to force the equation this way, and then as bicarbonate combines with the hydrogen ion, it takes hydrogen ions out of solution and runs the equation this way. So I can also take this away, and then these piles become bigger and everything runs downhill. So if we look at it this way, I can either take this away, decrease the carbon dioxide, or increase the bicarbonate. In both cases, I'm going to run the equation downhill, and this combining with this will take away hydrogen ions and reduce my hydrogen ion concentration, which means to increase the pH measurement for that particular solution. So I can decrease carbon dioxide by breathing out more, that's a lung effect, or I can make the kidneys can say, hey, I'm going to save more bicarbonate and resorb it, put it back in the blood, save it with the nephron, put it back in the blood, and generate less hydrogen ions that way. Increase hydrogen ions, we're going to go in the opposite direction. So I want to go this way, so either that means I have to decrease this to draw the equation that way, or I have to increase this. So I want to increase carbon dioxide. That means I don't want to ventilate my lungs as much. Hypoventilation is going to drive the equation this way and generate more hydrogen ions, which will lower the pH of the blood. Or I can decrease bicarbonate by taking it out of the bloodstream into the nephron and excreting it in the bladder. Don't resorb it, don't save it, don't bring it back and put it in the blood, but let it go out in the bladder. So you've got this thing here. The lungs are going to slow the release of carbon dioxide, so it builds up in the blood. Everything runs downhill, and this gets higher in amount as this dissociates there. Or I can lower this end of the equation by getting rid of bicarbonate, a kidney effect, and that'll leave more unpaired hydrogens, which will lower the pH. So another thing to think about, we said that the oxygen can be very elegantly released into the tissues by a simple mechanism. And we know that the amount of hydrogen ions will affect the stickiness of oxygen to the heme molecule in the red blood cell, in the hemoglobin. So when we're out in the tissues, we have a lot of metabolism going on. That's going to generate carbon dioxide and water. As we burn nutrients all the way in the presence of oxygen, we're going to generate carbon dioxide. That's going to push the equation this way. That's going to increase our hydrogen ions. That has the habit of making the heme less sticky to oxygen. So as this increases, there's an opportunity here to lose the oxygen in the exact place where metabolism is working the hardest. So the more CO2 you have, the less sticky that heme molecule in the red blood cell in the hemoglobin becomes and tends to release oxygen. So wherever you have higher metabolism, you're going to have a tendency for those red blood cells to dump oxygen in that location. And that to me is a beautifully elegant system to get oxygen exactly where it's needed. And if an area is not as active, we're not going to dump it off. But if it is really active, that is an opportunity to dump that oxygen exactly where it's needed. And I think that's pretty cool. So we know that red blood cells take up a massive amount of oxygen in the lungs with the use of hemoglobin and the heme molecule. The affinity for hydrogen is highest in the alveoli. It becomes very, very sticky because we're pulling CO2 levels out there, the lowest the blood will see, because we're ventilating that out through the lungs. The pH is relatively high in the small range that it does vary. Temperature is a little lower, but the CO2 levels are low, and that makes the oxygen very sticky to hemoglobin. So we've shifted the Henderson-Hasselbalch equation to the left, and we have less hydrogen ions, so things become very, very sticky. And then we talked about the release in the tissues. So at tissues where the oxygen is needed the most, hemoglobin loses its affinity for oxygen because we have a lower pH, more hydrogen ions. The muscle tissues are working, so you've got a slightly local higher temperature, and then because the muscles or wherever is working, we're generating more CO2 as an end product, and that is going to release the oxygen where it's needed the most. So loses affinity for oxygen under these conditions. So this is an active tissue. That means we're going to dump that oxygen where it needs to be. Another way to look at kind of the same thing. So you have oxygen here, you have oxygen and hemoglobin here stuck together. You have that hydrogen ion. It's going to knock the oxygen off. The oxygen is going to go to the tissue. We have respiration. That's going to generate more CO2. CO2 here is the Henderson-Hasselbalch equation. We have our unstable intermediate. We have bicarb and hydrogen ions. We have an exchange back and forth between the charges, making sure everything stays where it needs to be. These two, carbon dioxide and water, are turned into carbonic acid by Ca, which is carbonic anhydrase. So it's an enzyme in the body that promotes this formation and allows that there. But we have the same thing again, depending on what our levels of gases are in a particular area. That's going to affect the ability of hemoglobin to carry the oxygen, the affinity of hemoglobin for that oxygen. So pH, we have a very narrow range in the body. If we get much outside this 7.35, 7.45, we're going to probably die pretty quickly. So we average about 7.4. We got a couple points, a couple hundredths of a point in each direction, and that's about as far as we go. So acidosis, when pH is low, is a 7.35. We have more hydrogen ions present. Over here, that 7.45 is our alkalosis when the pH is high. So it's not a big range here. We're talking about 1/10 of a point. But that difference there is the difference between in the body acidosis and alkalosis. So acidosis here, alkalosis here. So when we're talking about regulating and making sure we don't go too far in any direction, we have that Henderson-Hasselbalch equation. How do we manage our bicarb or generate bicarb to buffer the system? So we talked about this pH here is going to be monitored by respiration, i.e. the lungs, and then renal function, i.e. the kidneys. So here's that slide we were talking about before. So are we dealing with acidosis or alkalosis? And is it a respiratory issue or a metabolic issue? So we start out with hyperventilation. We're going to get respiratory alkalosis. We're going to take that Henderson-Hasselbalch equation. We're going to get rid of the CO2 on this end and drive everything over here. We're going to lose hydrogen ions, and that's going to give us an alkalosis. So respiratory caused by the lungs, alkalosis, less hydrogen ions. In pulmonary edema, we usually have a respiratory acidosis because we can't clear that carbon dioxide as well as we might. So you're going to get a buildup of CO2 on this end of the Henderson-Hasselbalch equation. That's going to drive here and increase our hydrogen ion concentration, and that's going to give us an acidosis in the blood, and that's going to be related to the respiratory function of the lungs. We have compromised the lungs in some way, and we can't push out that lungs as well as we could. Repeated vomiting. If we're bringing up stomach contents, we're going to lose acid in the blood. That's going to leave us with a base overload, and that's going to be metabolic alkalosis. We've lost hydrogen ions by throwing up. We don't there. And then the other end of, if we go the other way in the digestive tract, the small intestine is not working what it should. We're going to lose bicarbonate. That's going to leave acid in the system, and that's going to be a metabolic acidosis, an increase in those hydrogen ions. So we have these situations here. The lungs is usually a short-term, very quick reacting thing. These can be short-term. The kidney over time is going to try to balance out what's happening. So in response to alkalosis, where we have not enough hydrogen ions in solution, we're going to let the bicarb go. That's going to drive the Henderson-Hasselbalch equation to the right, and that's going to mean we've got we're going to have more bicarb in the urine. That's going to increase its pH, and we're going to have more hydrogen ions because of the Henderson-Hasselbalch equation. So hydrogen ions go up in the blood, therefore the blood pH decreases. So the kidneys are working to counteract these two alkaloses. So we need to promote hydrogen ions decreasing the pH of the blood. In response to the two acidosis, hyperventilation is the first example. We may do some hyperventilation with acidosis, trying to correct that in some way, and then we bring more bicarbonate back in to the blood from the nephron, the proximal convoluted tubule, and that's going to increase the pH by driving the equation to the left, the Henderson-Hasselbalch equation. So bringing down the hydrogen ion concentration, putting more bicarbonate into blood, that's going to shift the equation, reducing the hydrogen ion concentration, and therefore increasing blood pH to counteract that acidosis. So you can have two different things. The lungs and the kidneys may be working in opposition, and that can be a challenge or a stressor on the body as you go forward. So that's the end of blood. Again, we're focusing on that Henderson-Hasselbalch equation, and we're trying to drive the equation in two different directions. We talk about either lowering one end or increasing an end, and everything runs downhill. So if we run to the right side of the equation, we increase our hydrogen ions, which increases or decreases our pH, makes it more acidic. If we increase our bicarb or lower our carbon dioxide, we run everything downhill to the left, and that decreases our hydrogen ion concentration. As we move forward, we get toward the end of the semester, we're going to be talking about metabolism. I'm going to be using in our drawings 3 colors. I'm going to use black for parts and pathways when we're comparing things out. I'm going to use red ink to denote hormones and their action. I'm going to use green ink to denote nutrients that are flowing through the system. And then I'm going to use blue ink to denote the processes and metabolic pathways. I'll use single letter functions by the time we get there to talk about G is for glycolysis, P is for protein prep, F is for prepping fat. So if you want to follow along with those colors, have them probably in 2 weeks. So you can follow along. So that's corner of the side. The nervous system. So what is it? We've got a collection of tissues that are able to respond very quickly to a number of different things. There's electricity involved. There's a very definitive organization and a very definitive set of pathways that an impulse follows. So we'll talk about how all that comes together. First, we'll talk about how this system is organized, and then how it works. So we're looking at the nervous system. We're going to deal with 2 different parts. First, we have the central nervous system, and we'll use the abbreviation CNS to denote that. And that's kind of the center of everything, the central nervous system. And then you can probably guess, if you haven't heard of it before, we're going to talk about a peripheral nervous system, PNS. So you've got the stuff associated with the axial skeleton is going to be our nervous system. And then everything outside that axial skeleton is going to be our peripheral nervous system. So here we're kind of looking at both our central and a peripheral nervous system. We start up here with the brain, as we saw in our fold, and then we've got the spinal cord going down. Anything inside, more or less, of the spinal cord, or protected by the spinal cord and the cranium portion of the skull, we'll consider the central nervous system. If we look at all these other major pathways that are very organized, very definitive, as we look at our different sets, we have cranial nerves here associated with the cervical, or not cranial. Cranial are part of the face. We have cervical nerves that come out of the neck region. We got the thoracic nerves until we get to the end of the ribs, then the lumbar, the sacral, and then we got a few associated with the caudal range. So we talked about the vertebrae being conserved in number. They don't vary much in mammals because we have to have this wiring system certainly from animal to animal, has to be consistent. Where do our nerves come out that control our front legs? Where do the nerves come out that control our back legs? Those things are not something we can adjust from generation to generation. They have to be more or less consistent. Another drawing, starting to label some things. So you've got the brachial plexus, that's associated with the arm. You've got the radial nerve, the ulnar nerve, depending on which one's going there, median nerve, femoral nerve, sciatic, running down pretty much the entire leg, and that coming out of the sacral portion there. If something pinches, that can be very painful. Lumbar sacral plexus, kind of sitting in between those two sections of the spinal cord. Brain up here, spinal cord through the vertebrae. Another look at the same sort of thing. Each diagram's got its strengths and weaknesses, but you can see as we go farther and down the spinal cord, we have pathways coming off in a number of different directions. So taking care of the thoracic cavity, going off either side. We've got rear legs, front legs, head, facial nerves, all working together under the coordination of the central nervous system. So we have all these nerves, and this is sort of the basic of what we're thinking about, especially if we're thinking about the voluntary system or the skeletal part. So we have here looks like some skeletal muscle. So we talked about one nerve. So this here is the body, cell body. We got our axon here, and then our nerve endings down here. We've got the dendrites here, we've got the axon here. The flow of information is, in this case, left to right. So direction of the nerve impulse, we start up here on the outside of the cell on the dendrites. If there is enough stimulation to those dendrites, there will be a signal fired down the axon, and that may or may not stimulate the muscle fibers depending how strong they are. There's two divisions to the nervous cell, as we mentioned already, the central nervous system and the peripheral nervous system. The central nervous system in this diagram we can see here is the brain, spinal cord, and then basically all the things that are protected by the axial skeleton, by the skeleton and the vertebra. The peripheral is everything outside that central nervous system. And when we're thinking about the peripheral nervous system, we have two-way communication. We'll have that too in the central nervous system, but we need to think of things that are sensing and things that are sending signals out to invoke an action. So the central nervous system, as we said, brain and spinal cord, housed in what part of the skeleton? The axial skeleton. The central nervous system is responsible for integration. It's going to listen to the inputs. We call them afferent nerves. Put the inputs in. The central nervous system decides what's going on and then sends an answer down efferent nerves. So some sort of response. We take the inputs, we integrate them, we decide, we yes, no, whatever it is, and then we send an answer, whatever decision's been made, we send that answer out into the body from the central nervous system. So central nervous system, we've got the skull of a horse here. So we talked about, you've got the cranium up here, and then these are all facial bones, the mandible down here. We did this with our foal. We sliced the head with that sagittal section, more or less on the midline. So central nervous system, the brain is up here, and that's going to exit out into the spinal cord, and that's going to move down through the top portion of the vertebra. There's a canal in there that's going to allow all that to move through. So this is a very detailed discussion of the brain and all the different parts here. This is the Atlas of Equine Anatomy. These are way more than we need to know of what's going on here, but let's talk about the forebrain up front here, the midbrain through here, and then we've got the hindbrain here. And we'll talk about those parts as we move forward here. So this is a good separating picture here with the colors. We've got the forebrain in gray, all the different parts associated with that. We've got the midbrain in through here. We got the pituitary and hypothalamus down in here. And then we've got the hindbrain with its different parts here. We could see very clearly in that fold the forebrain and the cerebellum in the back here. We kind of lost the spinal cord, but you can see that that's all part of what leads to the spinal cord. So you have more thinking and reasoning function up here, and then a lot of what's done down here is more or less automatic. There's patterning, there's reflexes, there's things we don't think about, heartbeat, lungs, kidneys, all those things are regulated and managed through the hindbrain. So if we had done a different way of opening our skull in our dissection, where we kind of sawed around and then took a lid off kind of thing, we'd see these membranous outer covering of the brain and spinal cord. These are the meninges. So it's kind of that tissue there. We didn't really look at that too much when we were doing our dissection. And then underneath that layer, you have three regions, as we mentioned: the forebrain, the midbrain, and the hindbrain. So here are a lot of the different parts. So we're looking at a lateral view here. This is probably pretty similar to what we did in that dissection. So you've got the olfactory bulb here, you got the optic chasm here that connects to the eyes, you got the hypothalamus, the pituitary. I think that looks like it's cut off a little bit in that picture, or covered up on the overlap here. We've got the forebrain here, the midbrain is through here, the cerebellum, and then the lower parts of the brain down here, the pons, the medulla oblongata. So we have the olfactory bulb here, and then we're looking underneath the brain here, kind of flipping this over this way. So this is now the front of the brain, the optic nerves going to either eye. You got that optic chasm that we saw before, so they're all connected there for sight. We got the pituitary underneath, and then we got the hindbrain with the pons, the medulla oblongata, and then you're going to transition into the spinal cord. Another good diagram here kind of showing those parts. You've got the cerebrum up here. You've got the hypothalamus through here. And then you got the thalamus through here. You got the pituitary gland. So we have a connection between the hypothalamus, which is nerve tissue, and then we have the start of the endocrine system coming out of the brain in the pituitary gland. You've got the midbrain kind of sits in this dividing area here. You got the pons, the medulla oblongata, and then the cerebellum here sitting kind of behind the brain. This was very obvious in our sectioning of the head, and this was as well. So the forebrain. Cerebrum up top, that's classically what we think about the brain, that dome that's got a lot of grooves in it. That's going to generate motor responses. It's going to give us some of the higher thoughts, thinking, reasoning, possibly emotions as well, and certainly in humans, maybe something a little bit more rudimentary in a horse. We have the thalamus, which is going to sit right above the hypothalamus, which is right here. And then underneath that is going to be the pituitary gland. So the relay center for voluntary actions. So we make all these decisions in the higher brain. They're going to come down through the thalamus. You got some hypothalamus does some involuntary stuff. It's again connected to the endocrine system, and it can direct, the hypothalamus can direct the production and or release of hormones from the pituitary gland. So depending on, we're dealing with the anterior posterior, we'll get to that next week. So thalamus is going to be sitting up there. This is through the things that go through. Everything kind of goes through that on its way down the spinal cord. So higher order thought is processed, and then it moves down, hopefully out and through. So in this case, we're looking at, you've got some sort of sensor on the skin that's going to come up and then be processed through the thalamus into the higher brain. But you got two-way communication going through the thalamus, stuff coming up, stuff going down. At midbrain, we're going to have a relay center for many of the involuntary actions and circadian rhythms. So we've got the midbrain in this section here. This is kind of an exploded version of it, our view of it. You've got a lot of patterning and sorting of thoughts and ideas, getting down to the hindbrain. So the cerebellum, fine-tuning of balance and movement, relatively large in a horse. Somebody wiser than I suggests that's maybe why a horse is so agile and can do so many different things with its leg and its feet and its balance. It needs that extra ability there in the cerebellum, in the hindbrain. The medulla oblongata and the pons, they're the conduit between the brain or the spinal cord and the forebrain. So you've got the spinal cord coming out of the medulla oblongata there, and then you've got the thalamus, the forebrain, thalamus, working your way through the midbrain to get to that lower brain. So you've got a lot of things here we don't think about. You've got heart rate is controlled here, respiration rate, body temperature. We've got the coordination of gait movements with our horses. So you could, in theory, take away the forebrain and they still would know how to trot, they still would know how to gallop, they still would know how to canter because of the patterns that are saved or initiated from here. I like to think about it like I brush my teeth in the morning. The brain says it's time to brush your teeth. So that to me is the upper brain. And then I've patterned the way I brush my teeth that I know that I'm going to start on my left side. I'm going to work my way around, and I make sure I get nice and high because of the way my wisdom teeth were taken out, make way high in the back. I come down, I work my way around on the lower teeth. I don't think about that pattern. It's just the way it is. I've done it so many times. I can think about other things. I can do other things while my lower hindbrain is working through those movements. Same thing, like coming out of the shower. Your towel, I tend to do the same pattern over and over again. Do this arm first, work my way from my hair to my toes. I don't think much about the pattern I'm doing because it's kind of into my almost unconscious lower brain, gives me a pattern. that gets me through. So when I probably first did my towel, it was more like, well, I need to do this arm, now my face, my head. Whereas now it's just, I need to dry off, and we've taken that script and programmed that into things we're not thinking about consciously so much anymore. Same with running or all those other things that fit in there. Brain says run, the pattern is pulled out of my hindbrain. We get to the spinal cord in the back. We have a situation where things are coming into the brain. We think about the dorsal root, I believe. We come in, and then we go out through the ventral root. So we've got a dorsal root. If this is the top of the horse, this is the belly or gut down here. Things go in this way and come out that way. So we're trying to keep things organized, trying to keep the highways working, and we've got two-way communication. Information comes into the central nervous system, information goes out of the central nervous system. So we collect information in the peripheral nervous system, we send it to the central nervous system, and then we send it back out to the peripheral nervous system. So the spinal cord is going to do a lot of receiving of signals, and it's going to distribute signals. They come in. Does it need to go to the brain or not? Do I just need to, it's a hot plate, I need to pull my hand away? The brain doesn't need to know about that. The spinal cord can take that reflex and take it through. So if you look down here, we've got our vertebrae there, we've got the cartilaginous connective tissue disc, you've got the spinal cord going through the top, and then you've got a nerve root coming out on each of the vertebrae. So a specific root comes out between two specific vertebrae in a standardized and recognizable pattern that if we're like dealing with nerve blocks and things like that, we know exactly which nerve I have to make the leg numb in the front, the leg numb in the back. I know exactly which ventral root I gotta get and dorsal root to stop that nerve transmission. So we're looking at our vertebrae here going through. This is the top of the animal, this is the gut, the ribs going down. The spinal cord's going to go through this lighter area here. Between that there, you've got the hole in each of those vertebrae. You can see that right there. So this is like a cranial view of the vertebra. This is a lateral view looking at it from the side. So seeing how it goes through the canal that's formed there. And we have exits or nerves coming off it at specific locations. If we get a pinched nerve, we got a herniated disc, somehow some liquid or some tissue is pushing up against a nerve as it comes out, that can cause real problems because we've got additional pressure on that nerve as it comes out of the spinal cord, and that pressure is going to cause problems. I'll skip that. So peripheral nervous system, everything outside of the skull and vertebra. These are the communication lines between all the parts of the body and the central nervous system. We have nerves going up to the spinal cord, the central nervous system. We got nerves coming back the other way. So you have a sensory or afferent side that's going to collect information about the world as we perceive it. And then you have a motor side where the central or efferent side, where the central nervous sends out commands, tells the body what to do. So we can think about moving our voluntary muscles or making some adjustments with our viscera in some way, shape, or form. So that peripheral nervous system, we're looking at an animal We've got cranial nerves up here. We've got spinal nerves coming out, and then we have those spinal nerves are inside the column here, and then when we leave the spinal cord, we're going to have parasympathetic and sympathetic nerves that are going to be our motor nerves. So when we think about the central and peripheral, what might the similarities be and what might the differences? So this is a Venn diagram where we have, say, this is the central nervous system, descriptors of that. And then we have this circle over here is the peripheral nervous system. So over here are things that only happen in the central nervous system, and over here are only things that happen in the peripheral nervous system. But there are some commonalities that we've discussed between the two. And so how are the central nervous system and the peripheral nervous system the same? And then how is each different? Give that some thought, and it will be part of your study guide. So we're looking at neurons that make up the nervous system. These are the specialized cells that transmit these signals back and forth very, very quickly. We're talking milliseconds to traverse from a toe to the head and vice versa. So neurons are the cells of the nervous system. If you go over to the campus store, you can buy a neuron there. It kind of looks like a funky looking lion / elephant. You got this face with kind of things sticking out of it that sort of looks like a lion's mane, and then you got this long trunk coming down. So the lion's mane is the dendrites, the face is the body of the cell, which has all the things we'd expect, and then you've got an axon in a generic nerve cell that runs out of the body of the neuron and moves things there. So the body, as we said, has all the nucleus, the cytoplasmic organelles, all the things you'd expect in a generic cell. But we have these extensions from the nerves, some quite long. So you have some nerves that originate in the neck and will go all the way down to your fingers, deep into your cavity. So we're looking at things that are feet long. So long, thin extensions. You've got the dendrites. Those are the kind of lion's mane hair thing we were talking about. And they're going to receive signals. They're generally shorter. And then, so that's going into the body. And then coming out of the body, you have the axons. They're going to send signals out from the nerve. They are longer, and they're covered by myelin sheaths. We'll show you what those are in a minute, but they are insulating the nerve. We can think about it as a plastic on a wire. We need to protect those electrical signals, so we have these myelin sheaths that are going to insulate the axon as it sends a signal. So here are several neurons from our Anatomy and Physiology of Farm Animals, 8th edition. We got a fairly standard neuron here. This is the one I described at the campus store. We've got the kind of lion's mane up here. Those are the dendrites. We have the cell body, which is sort of the face, and then we got the long trunk-like thing. That is the axon that comes out of the neuron. So depending on where we are, depending on what we're doing, we can have a couple different things here. Again, we have a main body that's not really different than the generic cell we talked about, but you have, let's see, we've got a bipolar nerve here. These neurons are characteristic here of this, the sensory neurons. We've got something sensory here as well, the unipolar. So we're going down here, but this is a fairly generic thing, and you're going to see this shape as we go through. So here's that neuron, the one on the right there. We've got the dendrites up around here. We got the nucleus, all organelles. So the dendrites are going to take signals from a number of different sources. Then they're going to decide whether they're going to fire or move things out. And then we have our axon here that's going to hit one or more other cells at the other end, and then the next cell decides what to do. So we have the axon here. It's got our myelin sheath on it, and what that is made up of are called Schwann cells. These Schwann cells will wrap around the axon, and because they're made of fat, they are hydrophobic, so they protect or don't allow or conduct charges. We're made up of hydrocarbons, so we know they're nonpolar, and they're going to protect the signals that are generated in the axon. Another diagram of a fairly standard nerve cell. Again, body of the cell here, dendrites, signals coming in from all directions. This cell decides, yes, I will fire, no, I won't. So you've got the myelin sheath made up of individual Schwann cells. And you can see here, we've got that cell that wraps around and around and around and around. We've got that plasma membrane. We've got the cell filled with fat, and that's going to act as insulation. That Schwann cell is going to wrap around the nerve axon and allow that cell to be insulated. So this is what we'll see during the time of the peripheral nervous system as it develops. You're going to have the axon in the middle, and then you're going to have that cell wrapping itself around to form that myelin sheath, the individual Schwann cell wrapping around the axon over time. So that myelin there. So another drawing of the same thing, or a picture actually, a micrograph. So Schwann cells wrap around to make the myelin sheath. So here's a cross section of a nerve. You've got insulated, so it can carry impulses much faster than non-myelinated. And that's part of the signal jumping from one side of the Schwann cell to the other in what are called the nodes of Renvier. So if you look back, you'll see those things, and the signal tends to jump from one node of Renvier to the next. I'm not exactly sure how that happens, but we know it does. So here we go. We've got some nodes between here. You've got two cells in sequence. You've got the axon of this cell hitting the dendrites here. You've got other axons here. And then this postsynaptic neuron has to decide, based on input from here, from here, from here, from here, from here, whether to fire and send another signal. You've got also example up here of a neuron sending signals to two different areas, and there can be some reasons for that as we need in the body. This is an example of a receptor cell. So you've got probably a touch receptor or pressure receptor down here. You're going to get a certain amount of pressure on that. The more pressure you get on there, the more likely it is to fire and send a signal up that axon and into the central nervous system. If you don't push enough on here, you're not going to get much of a reaction. It'll start but won't get too far. Once it gets going, we hope that an action potential moves all the way into the central nervous system.